Good morning, students. Today we are going to discuss DNA mutation, which is part of your Unit 4. Okay, so here we will say what are the different types of mutation and how does it happen. So before we discuss DNA repair, we will see how the mutation happens. In the background, we will be covering central dogma of life, and then we will see and try to find out at which step of central dogma mutation occurs. Okay, so we will see, is it happening during translation, that is at protein level, or is it happening during transcription, that is at the RNA level, or actually it is happening at DNA. Okay, so I am sure you all are already aware about central dogma. That is, DNA gets transcribed into RNA, and then RNA gets translated into individual amino acids, chain of amino acids, and after folding, it forms the mature protein. Okay, so here we are assuming that the DNA sequence, the stretch of DNA is ACAGGA, which after transcription forms UGU-CCU, which will eventually form the amino acid cysteine and proline. All right. So, but if everything is not correct, then mutation happens and the protein which is being formed is mutated. All right. So, here I will explain to you about mutation with an example of sickle cell disease. So, as I said before, that we will check all the three levels, protein level, RNA level, and DNA level, and see and try to find out at which level mutation is happening. So, in case of sickle cell disease, there is a protein in RBC. You all are already aware, I think, hemoglobin, right? HB, which coordinate with iron, Fe3 positive, to hold oxygen and transport them throughout the body. Mutation that causes sickle cell disease result in mutated form of hemoglobin. Okay, so where HbS is formed instead of Hb. Okay, so the difference between Hb and HbS means the mutated form is formed because glutamate is being replaced by valine. That is what is happening in case of sickle cell disease. This mutation results in HbS cells aggregating in RBC, that then it becomes quite difficult for RBC to transport oxygen effectively. Okay, so that is what actually happening in case of sickle cell disease. So now you all know that RBCs are generated by hematopoietic stem cells. Okay, and now we will see at what step of central dogma, the three steps, the central dogma, this mutation is occurring. So let us go the first one, okay, and that is at the protein level. So we are assuming that mistake is during the translation. So you see this picture where DNA is converted into RNA and RNA into protein. In the normal case, if you take DNA strand as CTC, GAG will be formed. Okay, so then glutamate will be the normal amino acid which will be formed. But everything is not correct here. Okay, so what is happening? What is our assumption is it is getting mutated. So then in case of mutation, instead of glutamate, valine is being formed at the protein level, because here we are assuming that mistake is during the translation. So instead of glutamate, valine is being formed, which will produce mutated HbS. Okay, but the point here to be noted is, if a mutation happens only at translation level, cell will produce only one mutated HbS. Okay, so what we are trying to say is, if it is happening at translation level, okay, as we are going by assumption that the mutation has happened at the translation level, but if that is the case, then cell will produce only one mutated HbS, only one HbS for each mistake. Okay, so and since cells are making enormous amount of hemoglobin, one mutated HBS protein might not have that big of an effect on the cell. So we can say that mistake during translation probably don't cause mutation like the one associated with sickle cell disease. All right, this is how we are crossing out the possibility of mistake happening at the protein level. That is the first one you see here. Then next two questions are still there. Is it happening at RNA level or at the DNA level? Let's see. Okay, so now if it is happening due to mistake at transcription, okay, so top one is again the normal case, then mutated one is at the bottom one, the CTC is getting converted into GUG instead of GAG, which is then it will code for mutated HBS, and that is because it is valine instead of glutamate. Okay, if this mistake occurred, the cell will only make a few mutated hemoglobin for each mistake. Alright. Since an individual strand of messenger RNA will only be translated a couple of times before being degraded. So we can say that mistake during transcription probably don't cause mutation like the one associated with sickle cell disease. So that eliminates the possibility of mistake happen you know during transcription, that is at RNA level. All right. So that possibility is also crossed out. So not at transcription, not at translational level. Now what level then? It could be happening. We are left with DNA level. Let's see. Finally, we look at the mistake at DNA level. So DNA instead of CTC, okay, mistakenly CTC is turned into CAC, then our corresponding RNA from transcription will be changed, and ultimately what we will see is a valine would be produced instead of glutamic acid. Okay. Now, since the DNA stores all of its genetic information, that mistake would lead to all future hemoglobin produced from that gene being mutated. Okay, so all normal hemoglobin will be mutated to HBS, that is mutated hemoglobin. So now we can say that mutations will usually result from mistakes in cells' DNA and not from the RNA or protein. Okay, all right. So what is it? Okay, now we have seen that the mutation is happening at which level? It is happening at DNA level. So permanent change in nucleotide sequence of DNA is called as mutation. Okay, and how it is happening, how it arises? DNA can be mutated either by substitution, deletion, insertion, or duplication of base pairs. And why we should be so concerned about it? Mutations for the most part are harmless except when they lead to alteration in encoded protein leading to malfunctioning of the protein. This can be lethal too. Because of lethal potential of DNA mutations, cells have evolved mechanism for repairing damaged DNA. There are basically four major types of mutations: base substitution, deletion, insertions, and duplication. So we will go and discuss one by one all of these four types of mutations. Okay, so in case of base substitution, one base is substituted by another, okay, and it is also called as point mutation. We have seen and discussed the example of sickle cell anemia where glutamate is being replaced by valine, which causes sickle cell. Okay, type of point mutation depending on type of substituted base. Okay, so depending on the substituted base, the point mutation can be divided into these category: transition, transversion, okay, and then depending on effect of mutation, what kind of effect it is bringing by getting mutated, we can characterize into three categories, that is silent, nonsense and missense. Let us see each of these one by one. In case of transition, it is very simple and straightforward. When a purine is substituted with another purine, okay, A is substituted by G, all right, or a pyrimidine is substituted by another pyrimidine, that is T is substituted by C or C is substituted by T, all right. So then it is called a transition. But in case of transversion, a purine, that is A and G, substituted by pyrimidine C or T. So purine and pyrimidine are interchanged in this case, in case of transversion. So the possible outcomes could be T replaced by A or T replaced by G. Same way, C replaced by A or C replaced by G. Then A could replace with C and A could replace with T, and G can replace with C and G can replace with T. Same thing, there is a pictorial representation of transition and transversion. So purine interchange is transition, pyrimidine interchange is transition. However, if purine is changing into pyrimidine and pyrimidine is changing into purine, all these combination can result to transversion. In case of base substitution, we are seeing the types again, and silent mutation. In silent mutation, the amino acid sequence encoded by the gene is not changed and mutated is said to be silent. Mutation is said to be silent. Okay, so here you can see in this example, at the DNA level, new mutation TTC, okay, in case of silent mutation it is TTT. So C is replaced by T in case of silent mutation. So the mRNA level will be AAG in case of new mutation, but in case of silent mutation that will result in AAA. But eventually, if you look at the protein level, both in no mutation and in case of silent mutation, they both are resulting in same amino acid. Okay, so no effect at protein level, and that is why we are calling it as silent mutation. So if a base substitution occurs in the third position of codon, there is a good chance that a synonymous codon will be generated, and that is what we call it silent mutation. In case of missense mutation, this type of mutation is a change in one DNA base pair that result in the substitution of one amino acid for another in the protein made by a gene. Okay, missense mutation is either conservative or not conservative. In case of conservative missense mutation, the structure and protein of substituted amino acid are very similar to original amino acid. Okay, usually have little or no effect on resultant protein structure or function. That is why it is called conservative. However, in case of non-conservative, the substitution leads to an amino acid with very different structure and properties, usually deleterious, very bad for resultant protein structure and function. That's what we have seen in case of sickle cell point mutation. So here is the diagrammatic representation of missense mutation. You see the original DNA code, okay, that is DNA bases, all are coding for amino acid histidine, okay, histidine. And then what is happening when replacement of a single nucleotide, that is nucleotide adenine is replaced by cytosine. You see the highlighted area in the second sequence, where adenine is replaced by cytosine in the genetic code, introducing an incorrect amino acid into the protein sequence. Okay, so instead of this histidine chain, now a new protein, that is proline, is being formed. So this is an example of missense mutation. Now again in base substitution category, we see nonsense mutation. When a base substitution results in a stop codon, ultimately truncating translation leading to non-functional protein. Alright, so you see the original DNA code and the amino acid chain respective to their codons from mRNA. Then what is happening here in nonsense mutation? The nucleotide cytosine, okay, is replaced by thymine. So you again check the highlighted region. So cytosine is being replaced by thymine in the DNA code, signaling the cell to shorten the protein. So what is happening because of introduction of that one nitrogenous base, the stop codon is being coded, okay, and that is what is happening. Because of this, the protein length is being short because it codes for stopping the translation. Next is deletion. It changes the number of DNA bases by removing a piece of DNA. Small deletions may remove one or a few base pairs within a gene, while larger deletions can remove an entire gene or several neighboring genes. The deleted DNA may alter the function of resulting proteins. Alright, let us see by an example. Alright, here you can see in this example one nucleotide adenine is deleted from the DNA. You check the top DNA bases, the sequence, original DNA code, and then you see the adenine in the next picture is deleted from the DNA, changing the amino acid sequence. Okay, so as soon as even just change in one nitrogenous base pair, it will change the entire amino acid code. Because as you remember, the codes go by 3 3 3. So one is gone, then the frame shift happens. Okay, the whole frame shifts that result in changing the amino acid sequence. So instead of histidine, histidine, histidine in top panel, you see histidine, histidine, histidine followed by leucine, isoleucine, and a lot of different amino acids. Then next is insertion. An insertion changes the number of DNA bases in a gene by adding a piece of DNA. As a result, the protein made by gene may not function properly. Let us see by an example. In case of insertion, okay, one nucleotide, that is adenine. Look at the highlighted area in the the second picture. The highlighted area is adenine is added in DNA code, changing the amino acid sequence that follows. Okay, so you check the amino acid sequence in the top panel, then look at the amino acid sequence in the bottom panel, and because of insertion of adenine, this is what is happening. Alright, so last one, duplication. Okay, a duplication, as the name suggests, consists of a piece of DNA that is abnormally copied one or more times. This type of mutation may alter the function of resulting protein. So this is what is happening, duplicated mutation. Okay, a section of DNA is accidentally duplicated when a chromosome is copied. All right, so that's all for today. Okay, keep studying. I have been posting the assignments on Google Classroom, so make sure you all go through the assignment and complete it on time. And also I will be posting my next video on DNA repair. All right, take care.